Greetings and welcome to ALAT Chapter 30, Fish. I have to tell you that fish are one of my favorite species. The fish that's up here in this very blurry picture because she moves so darn fast is a bloodmouth parrotfish, a type of cichlid. Her name is Peaches. She's like 5 inches big, and she is how big I am hoping my 3 bloodmouth parrots will get to. I have 3 because you aren't supposed to keep them in odd or even number groups. You need to keep them as either ones or fives or sevens because they will gang up on each other. Fish are very complicated, but so rewarding and just so fun. Now, we use several different types of fish in research, and they're often used to study endocrinology, bacterial diseases, even genetics. Toxicology studies, of course, can be done with fish. Research is also being done on nutrition, health, and husbandry of fish because of the importance of aquaculture or using fish as food. Tilapia is a really, really big one that gets researched. In addition, fish are often studied in their natural habitats because they can be used as a bioindicator, letting us know early on that there's something going on in their environment or ecosystem that is problematic. Peaches is so cute. So a little bit about the taxonomy. We are often using rainbow trout, catfish, and a variety of tropical fish species. The zebrafish, the Danio rio, is one of the most common aquatic species used in research. It is in the minnow family. We also use some animals in the trout and salmon family and catfish as well. This little 10 facts about fish research is from Understanding Animal Research, a UK group that does a lot of good educational materials. So I thought this would be really interesting because there's some neat facts in there. Okay, anatomy and physiology. Usually I have like 6 or 7 slides, it feels like, for A&P. We're only going to have a couple here. So fish are vertebrae, and that's important for us because it lets us study parts with parts. We're vertebrates, they're vertebrates, therefore there's good overlap. Most fish are ectotherms, so they're getting their body temperature from the external environment, and they have the ability to modify that temperature by moving either within the depths or just to a different area of water. This seems silly, but they have fins that help them move and gills which let them extract the oxygen they need from the water in which they swim. I feel like most of you probably knew that. They have scales. They also secrete mucus, and that mucus keeps their scale and skin waterproof and keeps them free of infections. They have something called an operculum, which is a bony flexible plate that covers the gills. It's also known as a gill plate or a gill cover. So it is here and also here. Kind of wanted you to sort of be able to see how big it is compared to the size of their gill. There is something called a lateral line that you can see on most fish. It's actually a sense organ, and that sense organ runs bilaterally, and it lets them feel movements and vibration in the water through their skin. And they also have something very, very important called a gas bladder or an air or swim bladder. Most fish folks call it an air bladder. And that organ helps the fish maintain its buoyancy and control its position in the water. Sometimes they have too much air in it, and you actually have to go in and extract some air out so that they can actually navigate as they would normally. When it comes to fish size, you can have tiny, tiny neotetras to giant whale sharks that are more than 20 meters big. If you've never been to the Georgia Aquarium to see the whale sharks, you have missed out. Granted, I would like to see them in real life. Our zebrafish measure less than 5 centimeters, so they're a smaller fish in general. You will need to know some stuff in here. So this is biological data specific for the zebrafish. You really will want to know their lifespan, how quickly they become sexually mature, if there's any spawning cycle. And then I really would pay attention to your water temperature and stocking density. you will be asked how many fish you can have per liter of water. 100%. So when it comes to sexing fish, some of them have sexual dimorphism, so you can look at the males and the females and you can tell which is which. But other species might not have any external differences, so the only way that you would know is if they have laid eggs or if you have had to do surgery. Sorry, still have this cold. Most fish are oviparous, which means that they lay eggs and rely on external fertilization. Sperm then gets released over the eggs after the female has released them. A key word here, spawning, that literally means the release of eggs and sperm. There are other types of fish that are viviparous, and they bear young, live young after internal fertilization. Um, viviparous, like vivariums. It basically, vivi means life. So that's why that term is associated with the fish that bear live young. Parental care is all over the board. Some build nests and take care of their eggs and take care of the newly hatched young. Others lay the eggs, spawn, and then they're gone. That's it. In fact, if they stuck around, they would just eat the eggs if they were still in the area. One helpful thing to know, females of live-bearing species are larger than males, but the male tends to be more brightly colored. The female egg layers, so the ones that are oviparous, ovi meaning egg, they will often have a swollen abdomen when they are ready to spawn. And this male and female zebrafish, you can see the male doesn't really have anything going on in the ventral area, whereas the female has just, there's just a little bit more hanging out there. Now, how fish get bred in labs really depends on the facility. But if you're working in general with zebrafish, which most of the facilities here do, you can breed many males and many females in a single tank or just a couple in a small tank. So it depends on what your space looks like. This is a really important thing. The breeding tanks usually have perforated inserts, which lets the eggs fall to the bottom while not letting them get eaten by the adults. And this is a really good schematic of what that looks like. So this is just a plate, plastic plate with holes. Female lays the eggs, male pushes out sperm, the spawning happens, but the eggs are physically separated from the parents so that they can't eat that tasty, tasty snack. I might have had sushi this weekend because of all of the fish talk. Typically, if you're doing breeding in a lab, you're going to put those fish together late in the day. The spawning will happen early the next morning. You can then collect the eggs and return the fish to their normal housing tanks. Things to know, the females used for egg collection are going to use a lot more energy than females or males that aren't being bred, and they need extra snacks to make sure that they can account for that extra energy. And those fish shouldn't be used more than once a week for egg collection. I told you to pay attention to your water temperatures. Water temperatures for zebrafish normally is about 28.5 degrees centigrade, but the embryos actually can take a wider range of 24 degrees centigrade to 33 degrees centigrade, so 74 to 80s. You can keep lots of embryos together if you keep them in an incubator or a water bath. This is a petri dish that's holding a whole bunch of them. And then those fry or young fish will start eating on their own 4 to 6 days after fertilization. And the type of feed that they'll need will change as they age. So where do we get fish from? Usually other labs or fish producers. And in order to ship them, just like in Finding Nemo, they will get shipped in plastic bags that are partially filled with water. Those are then placed inside insulated containers to make sure that they stay at a really respectable water temperature. The bags need to have a sizable air pocket to replenish the water's air supply, and you can see that here, this, you know, water here, and then all the air that they could muster in here. I don't know if any of you remember with Finding Nemo where the pufferfish was in the bag and then puffed and took up more of the airspace. Those of us in the fish world giggled real hard at that, knowing that he was also just going to poke holes in there and get loose really quickly. When you are handling fish, you want to make sure that you don't overcrowd the bag. This is actually, I think, too many fish for this bag. I probably would have gone half that many, but it does depend how far they are being transported. If you are moving them within your own facility, you can again move them in bags, or you can move them in coolers containing oxygenated water, or even buckets that have an air stone in them. When you have fish shipped to your facility, they really should get checked and unpacked immediately upon arrival because they are delicate. The conditions of the water in the receiving tank should really try to match the conditions of the water the fish were in prior to shipment. So hopefully you have that information, because putting them in water that's vastly different is very stressful for them. If you did receive fish in your plastic bags, and if any of you have ever brought fish home from PetSmart or Petco, you know that you really need to put that bag in the tank of the new aquarium so that their water temperature can acclimate. And then once that the bagged water has come up to or down to the temperature of the tank, you can transfer the animals to the tank using a net. You really don't want to put that transport water in the new aquarium. You just don't ever know what could possibly be coming with it. So generally speaking, you move the fish, not the water. Quarantine. If you're bringing in new fish, they really should be quarantined for about 30 days and need to be checked for signs of disease or any presence of parasites. Just like any kind of a quarantine, non-human primate, rat, mouse, you really want to have your equipment and tools dedicated to that area so that you don't accidentally bring disease into the existing population. In many facilities, the adult fish never actually move into the colony. They're kept in the quarantine, and they're used to produce embryos for the primary system. And those embryos actually get quickly disinfected in a bleach solution before going into the main colony. A workflow note: be sure that you're completing your work in other fish housing areas, and this is true for any animal area with a quarantine, before going into the quarantine. You don't want to go from quarantine back into your main colony. If for some reason you have to, you need to be sure that you're maintaining proper PPE. You possibly might have to change scrubs. And if you've been working with the fish colony, it's really important to wash your hands and your arms before you go back to work with the main colony. There are so many things that we can carry on our skin that could go from the quarantine area back to your normal colony. So fish behavior, I've got some video for you. I was really hoping to get a video of Peaches, but she was not cooperating as she does. She is a very funny fish. Fish normally are inquisitive and they're usually active. There are some species that hide, but most of the fish that we work with are usually going to be out and engaging. If they are in reproduction cycle or if you're feeding them, you're going to see differences in behavior. You'll also see differences in behavior if you have overcrowded the fish. If you see fighting, and that can happen, it's really important to find the aggressor and move them out so that the other fish can have some peace. So this video is just here to show you normal zebrafish. This is a really nice tank. I love the enrichment in it. the fish are moving around. They're up, they're down, they're using the entire water column from floor to top. They're moving all the way around. That is what you want to see. If instead you were just seeing the same species of fish just kind of hanging over in a corner, up at the top in a corner, that might indicate a health concern that might need to be reported. Handling fish is very tricky. It's very stressful. I feel like I talk about how stressful it is to handle fish. A lot of times, depending on what you're doing, you are going to need to anesthetize fish. And the way that we do that is with Tricaine S, MS-222. It's a very commonly used anesthetic. It's actually administered through the water. So you put the fish in a smaller container of water with the MS-222, and that allows the fish to actually be anesthetized. Once you've completed whatever you're doing, they get placed in normal water until they recover. Regardless of whether or not you're using anesthesia, you really need to take care to keep the fish moist by placing them on a wet towel or a sponge and putting them back into the water as soon as possible. You want to wear wet gloves, just like we were talking about with the amphibians, and handle the fish gently. If you have bare skin, you could scratch off the mucus layer and really damage the skin. To prevent cross-contamination, you want to make sure that you're handling cohort by cohort, disinfecting everything in between cohorts. So I have two really fun things for you. I wanted to show you what a fish surgery looks like. So I actually was able to find a puffer fish surgery that happened in Florida. And then we'll also talk a little bit about fish anesthesia. I found some really interesting video on that as well.
Meet Kirby, our porcupine pufferfish here at Gumbo Limbo Nature Center. This fish can be found in shallow, warm, tropical waters and typically feed on hard-shelled invertebrates like sea urchins, snails, and crabs. Porcupine fish have interlocking spines covering almost their entire body, which they use for defense when they blow up. They can blow up 3 times their normal size when threatened.
I'm Keith Herman. I'm the senior aquarist here at Gumbo Limbo Nature Center. Kirby came to us at the end of 2017 at about 3 or 4 pounds. We did purchase her from a collector from the wild. We did notice right away that she was blind in one eye. 2018 or early 2018, we had about a year of, we didn't notice that there was anything wrong. So we had a good baseline for what to look for. So when Kirby started acting oddly in early 2019, we took a very close look at it. All of a sudden, she was no longer using the whole exhibit space. She was kind of hanging out right where the water inflow is, which is usually classically is a parasite problem with fish. So we wanted to make sure that we got her looked at very quickly. We checked her for parasites and did a broad spectrum antibiotic just to make sure that she didn't have something we didn't know about. When neither of those things turned up any evidence, we looked to our friends in rehab and they helped us do a radiograph.
The radiograph revealed that Kirby had a hook in her intestines. The next step was a CT scan, which helped determine the exact location of the hook in preparation for surgery to remove it.
I'm Dr. Maria Chatham, and I'm the veterinarian here at Gumbo Limbo Nature Center at Centura Rehab. Kirby was the first and only surgery I've ever done on a fish. I've never anesthetized a fish before, so I told Keith he was in charge of that.
fish surgeries and fish procedures, we do use an anesthetic for fish. We use a very light dose to help give them, help calm them down a little bit, help them not perceive any pain that might be associated with what we're about to do. So we try to make sure that we are being safe with that animal, but also safe with ourselves.
Because she is a puffer fish, she has spines, and the spines are actually like bone. And so I had to cut through the bone with a bone cutter in order to even open her up to evaluate her intestines. It was the biggest challenge because I wasn't familiar with their skin and their spines, and I wanted to be really careful not to damage their spines too much. Once I got into her, it was more similar to other surgeries that I've done in an animal's intestine, so that wasn't as challenging to me. I just realized how tough pufferfish actually are. Don't know how that hook ended up inside of her. She was really well fed before she went into surgery, and she didn't eat for about a month afterwards, which was normal, but I felt good about it because she already had some good flesh on her. Surgery did take about an hour, and the recovery, took a little while too. But it was fascinating to me. We did find out when we had her in recovery that she was a female because she laid eggs, and we didn't know that, so that was pretty neat.
Currently, we are celebrating Kirby's 3-year post-surgery anniversary. She is doing very well for us. During surgery, she weighed 8 pounds. We're estimating her current weight about 15 or 16 pounds. I would say she's probably almost tripled in length and more than doubled in size.
Thanks for watching. Come visit Kirby in our Coral Reef Aquarium.
So some neat things there. I don't know if you noticed in a couple of the spots where they were keeping her on the table, they had the moist towels. They also were continuously flowing water gently over her mouth, over her gills, partially because that water was anesthetizing and partially to make sure she had a constant supply of oxygen. Really cool things. Okay, so a little bit of identification. You don't always need to have your fish individually identified. You can just track per tank instead, but sometimes you do need to know which fish is which. And in order to do that, you can do what they're doing here, which is a subcutaneous dye injection that is very minimally invasive. You can also implant tags. You can clip fins. You can use transponders. Or what I do with a lot of my fish is individual body patterns. So the parrot mouths that I have, one of them has black pigmentation around her eyes, and her name is Satsuma. I know it's her because of the black pigmentation. Here's the fun thing though. Blood mouths also, or parrot mouths also spontaneously change their color. So they'll go from orange to black pigmentation, spotted, and then back to full orange, which makes it really confusing when you come home from a vacation and you wonder, did somebody replace your fish? If you are using something like the implant tags or the transponders, you need to make sure that you're inserting those under anesthesia. With tags, you need to make sure that their placement allows for normal behavior and aren't placed in such a way that they could get tangled. Husbandry for the zebrafish is really interesting and kind of hard to just explain. So again, videos, you guys are so welcome. I did end up muting this for you guys because the sound of fish tanks is very loud after a while. Okay, and then another one that I wanted to share with you was on cleaning and disinfecting tanks, because it's again something that a lot of us don't see when we are not working with aquatic species.
At our facility, tanks get cleaned about once every 5 or 6 weeks. Here we'll go through the steps that we take when cleaning a single tank. First, we have to get our supplies ready to set up a new tank. We fill up a clean tank using fish water from the rack. We make sure to transfer all of the important labels from the old tank to the new tank, and then we can start transferring the fish over. When handling the fish, it's important to move slowly and carefully as to not scare, or injure any of the fish. Be sure to replace the lid on the tanks after you're done handling the fish because they will try to jump out and we don't want to lose any fish. Now that we've transferred all of the fish into the new clean tank, we can put this clean tank back up on the rack and start the water flow again, and we will move on to bleaching and cleaning the old tank. We start by cleaning any excess debris off of the dishes using a soft sponge or cloth. We can use tap water at this stage, and we will put them into the bleach. We make sure that all of the dishes are completely covered by the 10% bleach solution, and we will leave them in the solution for 30 minutes to disinfect them. After the bleaching period is done, we'll remove the dishes and rinse them under tap water. Bleach is very harmful to fish, so to make sure any leftover bleach is inert, we place the dishes into a sodium thiosulfate bath for a few moments. We will do one final rinse in tap water and place the dishes onto a drying rack to dry. If the dishes need to be used immediately, then they need to be rinsed with RO water to make sure that there is no chlorine left on the dishes from the tap water. So as you can see, this is a fairly time-consuming and laborious process. We hope to be getting one of these large cage washing systems soon enough.
Okay, some things that we wouldn't see if we weren't working with aquatics. So back to a little bit of the husbandry. As I'm sure you remember from some of the other conversations we've had, water quality is the most important factor in successful fish husbandry. There are a lot of factors that have to be monitored: pH, salinity, oxygen. Little changes in the water condition can actually have a huge impact on the health of the fish. Here's a list of some fish-specific factors that are really something to pay attention to. So disinfectants or detergents, don't use them because the residue can impact the health. Leftover feed and feces should be removed as quickly as possible. Salt deposits on the tops of the tank surfaces should be cleaned off as quickly as possible. Algae, biofilm, and just gross matter should be scrubbed from tanks if you're autoclaving those tanks before they go through the autoclave. If you are autoclaving tanks, do not autoclave the biological filters. Those were those little white wafers. Autoclaving those would kill the good bacteria and cause problems that could ultimately kill the fish. And then if you are adding a new fish cohort and you've euthanized the other or they've gone elsewhere, you want to make sure that the room and the housing systems get disinfected at the end of the study before bringing in a new cohort. Things that we already know: health evaluations of each tank needs to be performed daily. You really need to make sure you have eyes on every single one of the fish. Sick fish should get removed immediately because it's possible that they could spread disease to other fish in the tank. Depending on instructions from researchers or veterinary staff, those animals will either be quarantined or euthanized. And then deceased animals need to be removed quickly from the tanks because the carcasses can actually contaminate the water supply. Optimum stocking density, 4 to 6 adults per liter. And that is zebrafish specific because remember, size matters. These are small fish, so you can have 4 to 6 of them per liter. That would be different for a larger species of fish. So a little bit about fish diet. Some fish are herbivores, others are carnivores, and others are omnivores. If you are feeding carnivores, you're usually going to feed either a live diet or a commercially available dry diet. Live diets can be aquatic animals like Artemisia, rotifers, paramecium. They're usually raised on site and you have to take great pains to not contaminate them. Dry diets are so much easier. They are designed with the proper nutrition in mind. They need to be stored properly and used before the expiration date. In a lot of cases, a combination of live diet and dry food is used. And their feed needs are going to change according to the developmental stage of the fish. So if you remember the husbandry video where they had the pipette that they were pipetting out some of the fish food, that's because those fish were much smaller, so they had a different size paramecium being fed to them, or rotifers. Larvae need a lot more nutrition because they are growing, so larvae need more food than the adults. In general, zebrafish are fed twice a day, sometimes more. Please nobody tell this to my fish. Other fish species may be fed once or more a day. And depending on their developmental stage, they may need just a lot of food. So again, the larvae, the juveniles, they're going to need quite a bit of nutrition. This slide pains me. So ALAS, bless their hearts, their fish experts weren't quite as involved with this section, I think, as they will be for the revision. I can tell you because I'm already making notes of things that need to be revised. So we'll go with what the book says because this is what you will be tested on. The need for enrichment varies with the species. That is what the book says. I'm going to tell you that is the book answer. Enrichment may promote normal predatory and reproductive behavior. Providing hiding places or substrates such as sand are examples of enrichment that are thought to promote fish welfare. It is important to make sure that any enrichment that you provide has no negative impact on the water quality. That's a really, really big thing. And really cool, providing live food is considered a type of enrichment. Now, those are the ALAS answers. I will tell you that I present the same lecture to a different group and had to do more recent citation research, and enrichment for fish has actually evolved a lot in the last 10 years, which is very exciting. Some things that the pufferfish do, pufferfish are so funny. They will play fetch, they will play tag, they will go through hoops, and you can train some fish to do things like move balls into a net. You might think that this is a silly behavior, but it actually is similar to them hunting for the perfect rock that they could make a nest with. So it is a species normative behavior, which is the point of environmental enrichment, promoting species normative behaviors. Signs of pain, distress, or illness. I should have grabbed video of this. Bad Katie. So things that you might see, and if you remember the video of Kirby, they said that, she wasn't moving the same way. She was kind of isolating. She was a lot more lethargic. Those are normal things to see an ill or distressed fish do. Other things that they might do, feeding routine changes. So they might stop eating or they might start spitting out their food. They might make erratic movements like flashing, which is literally scraping against objects in the tank. They'd mentioned that they thought maybe she had parasites because she was up against the filter flow water. And you'll see that where they'll be rubbing up against the filtration equipment. Isolation, lethargy, piping or gulping air at the surface of the tank, and then swimming in circles, and not necessarily circles like in normal body position, but they could actually be swimming in sporadic circles, figure eights, looking very disoriented. You might also see their skin change, the condition of their skin fins or gills change. Their fins may actually clamp down. They may not be able to control their floating. That's that air bladder again. The operculum could be flashing or moving very rapidly, and that's because they're having trouble breathing. Remember, that's the gill cover. If you see anything like this, those fish need to be looked at by a veterinarian. Again, they should minimally be removed from the tank and placed in isolation. And the first thing that we look at anytime there's anything weird with fish, check the water quality of the tank. Sorry, I'm reading. I'm like, why is my fish sedation not in here? I wonder if I ended up not including it. Weird. When you are anesthetizing a fish, you can use the exact same thing that you use to euthanize a fish. It's that buffered tricaine MS-22. When you are euthanizing, you're basically going to like the fifth plane of anesthesia, which is where death occurs. So anesthesia and euthanasia are often using the same materials. When you are euthanizing fish, they go into properly prepared euthanasia solution. You really want to use the same water that is already in their tank so that they don't have to experience any stress from difference in water. Euthanasia of amphibians, reptiles, fish, it's not quick. It can take over an hour for fish to go from anesthetized to deceased. A lot of times, physical methods can be introduced, such as hyperthermic shock using ice. If you do that, the animal can't physically be in with the ice. They go into a container that's rapidly chilled with the ice slurry. Individual institutions might require other things, such as pithing, which is basically destroying the brainstem, or decapitation, to make sure that the animal is actually deceased, because the worst thing is to think an animal is and you put it in the freezer and then it's not quite as deceased as you thought. And anytime that you're doing fish disposal, because you're using a external substance, you need to make sure that you're doing it according to your facility guidelines and local municipality guidelines. You will see as you're looking at these slides that some of them have added materials beneath them, just some different references for you to take a look at should you have some extra interest. Okay, as always, I hope that was helpful. Thank you very much.